Welcome to Brain Pods. This is the first volume of what is going to be 13 sets. This is Dr. Chafee with me. You will remember him from the Med Neuroscience class in first year. Right now, it is the time of year where you are just halfway through, almost a little,
We are.
about halfway through.
Yep,
45 lectures.
that's right.
I
A
have-
few fewer this year,
I have
which you have
I
gone
have,
over.
I have the pleasure of looking through all of them. What we're trying to do here today is to provide a little bit of an introduction and review to neuroanatomy as you're about ready to start your third year clerkship. So you've done the foundational years, both neuroanatomy as well as the what is now called the HHD5 course, where you're introduced to neuropsychiatric disorders. Here we're taking that foundational knowledge and then expanding it into clinical management and the investigation and treatment of these conditions. So I'm thankful that you're here, Matt.
Oh yeah, my pleasure.
So the part of this is part of a larger experiment to integrate
Yeah.
what you're doing and what we're doing,
Mhm.
and we're going to try to summarize all 45 of your lectures down into one. So here we go. So if you're about ready to take step one, this may be useful. Or if you just took step one, you can send me an email. Let me know what else we need to include. But this is our attempt to do that. So let's just go ahead and get started. So just to state the obvious, clinical neuroscience is fascinating. Inside your own skull, you own the most complicated system in the known universe. 100 billion neurons, 100 trillion synapses. It is ever changing. Your brain, after watching and listening to this, will be different than when you started. I think that's just the inherent fascination that is the human brain kind of gets us going.
Absolutely, and yeah, and that was something we picked up on that in the course frequently, and of course it has large clinical relevance. It's just this idea of plasticity being built into the system at just about every level, and that in response to disease states or damage, the system can reorganize, and that I'm sure is a huge part of clinical practice.
The clinical neurosciences are the breakthrough specialties of the 21st century. What is going on in the development of neuroimaging, gene therapy, lytics, neuromodulation is very exciting. I will not wear you down with clinical cutting edge examples at this time. Those of you who are interested, just Google or YouTube a couple of the examples we laid out here. So stroke, when I started medical school, we were just beginning to do thrombolysis for infarcts. So when a stroke happened, it was, we were alive long enough to remember when if you had a stroke, there was nothing that could be done.
Yeah.
Except advising people to control blood pressure and stop smoking so that they didn't have another one. That is really no longer the case. If you really want to be inspired, YouTube SMA gene therapy. So SMA is this devastating anterior horn cell disease which can affect children. It is in the differential of the floppy baby syndrome. It apparently is a fairly simple genetic mechanism, and they can put RNA interference. And we already have, I just talked to our pediatric neurologist, Peter Karachunsky. We already have 40 patients on it, and in his very understated Russian way, he said, yes, they're doing well. It's just, and it's just, I mean, like, we're curing SMA now. I mean, that's just incredible. I'm getting back to studying multiple sclerosis because I need to retake my boards this year. And multiple sclerosis management has just completely changed from 10 years ago in terms of all the ability that we have in terms of helping to prevent attacks, flame ups by the immune system. And this last one I just put in here because it is something I'm just fascinated by. My colleague down the hall in psychiatry, Kelvin Lim, does neuromodulation, transcranial direct current stimulation, and he just blew me away the other day by showing me some of his data for people who've had recalcitrant alcoholism. The idea that you can, by enhancing plasticity,
absolutely,
you can essentially increase the executive brake on the nucleus accumbens, so that people with alcoholism feel less compulsive urges to drink alcohol.
Yeah,
I was like, "Are you kidding me?"
Yeah, there's a lot of work in this area that Calvin's involved in, but also a lot of new work in addiction. People studying animal models of it and being able to sort of dissect cortical striatal circuits and their influence on reward and reward behaviors.
Certainly a big impact on the opioid crisis, others as well. All right, that all being said, what my goal here was to approach all of the clinical tracks that you have taught our students, you and others have taught our students, and instead and to think about those in the way that we do the exam. So when we do the exam, we literally do it from the top down: cognition, cranial nerve testing, motor testing, sensory coordination, gait, reflexes are in there, and so that we're going to go through the pathways that are most relevant as you're examining someone. So that's how we're going to go ahead and take this, and we're going to start with alertness, because you can't. The very first step of evaluating someone's cognitive function is whether or not they're awake, and whether or not they're awake is related, of course, to the reticular activating system. And so I'm a sleep person, and so maintaining wakefulness has always been kind of an interest of mine. Are you familiar with how Maruzi and Magoon discovered the reticular activating system?
No, I'm not actually.
Well, so the Maruzi and Magoon discovered the reticular activating system. It was post-war, late 1940s, and they were doing stimulation studies on the feline model in cats. The reticular formation was long. I mean, it's been known for a long time. It's just it's a pathological term to describe that region of the brainstem, and they were looking at, if you would stimulate it, what it does to movement, axial posture.
Right.
And not long before that, within a generation, they had developed electroencephalography. So they were putting EEG on the cats' heads, and they were anesthetized, and then they were stimulating the reticular formation. And they were not expecting to see any changes. All they were doing was just confirming that the cats were anesthetized. And they stimulated them, and guess what happened?
They wake up.
They woke up.
Yeah.
And it's just like, oh my goodness, what did we just discover here? So the reticular activating system is a complex set of neurotransmitters. We will go over this in detail in Neuropods, the volume where we do the chapter on sleep. But there's histamine, dopamine, norepinephrine, serotonin is involved. In general, you can just think of it as a series of nuclei here in the reticular formation that send signals up to the cortex to help it wake up. Understandably, there's afferent input to the reticular formation from the extremities, i.e., if you get pinched when you're sleeping, that pain signal will not only go up into the ventral thalamus and the cortex, it'll also go to the reticular activating system to help you wake up. Noise, right? Auditory inputs will feed in there.
Right. Yeah, and one of the interesting aspects of this is the way that the system can modulate thalamic input to the cortex and help thalamic relay neurons switch between modes where they generate rhythms that are associated with sleep, versus putting them into a mode where they faithfully transmit information from the periphery up to the cortex.
Right.
So that's that sort of control of the thalamic gate for wakefulness is also a component of this system.
Right. So, in my understanding, is the reticular activating system helps shut down that thalamic relay. How would you characterize it a little bit more astutely?
During sleep, thalamic relay neurons enter a burst mode where they basically go offline, and they are not sensitive to external input anymore. They generate intrinsic rhythms that are sort of associated with sleep. But then the reticular activating system can help get them out of that mode into the tonic mode, where they're able to transmit information from the periphery up to the cortex for sort of processing and behavior.
Very cool. And then we wake up.
And we wake up.
All right. So now you're evaluating someone who is awake, and you would like to get an idea of what their memory is. So there's a lot of different circuits that you have probably looked at in various review books, in lectures, in terms of how the brain consolidates memory. This complicated circuit right in front of you is an incredible simplification of an incredibly complex process.
Yeah.
A couple principles just to remember is that there is a reason that the circuit for memory is tied in together with the circuit for emotion. And that is because emotions are our nervous system's way of assigning salience to experiences, right?
Yeah, that's right.
So there's a reason why your top 5 most embarrassing moments of your life, you can rattle at least I can.
Yeah,
I can rattle.
Hard to stop at 5.
Yeah, fair enough. But you can rattle them off because your limbic system works to essentially make sure you remember these, because human beings are social animals. It's really important that we interact well with each other, and when you're humiliated, that is your brain's way of essentially telling you, try to avoid this situation again in the future. But it's not just social cues and interactions. It's joy, humor, fear. Great way to remember things, as if it was associated with something terrifying. And that's what. And so essentially, when I think of the limbic circuit, and you can look at the circuit here, it is an interaction between how our brain consolidates memory in the hippocampus before moving it to the cortex, and how the limbic system in the amygdala essentially assigns salience to it. Any other thoughts to add on that?
Yeah, I mean, I think that's accurate portrayal. I think when I think of this system, one of the interesting things about damage to the system in sort of classical cases like HM is that the amnesia.
HM.
Yeah, HM. Henry Molaison was a patient that had bilateral surgical resection of medial temporal lobe to manage intractable seizures.
Okay.
And this is a very, very famous case, because following this surgery of destruction of the structures you see in the slide, couldn't lay down any new memories. Old memories largely spared. It's anterograde amnesia. It's the inability to sort of record new experiences, and I think that is a key sort of clue about what the system does and how it operates. It's sort of I think of it as being kind of a mechanism for taking a snapshot of events or episodes, and it does this by having very rapid plasticity within it. So long-term potentiation, which is the most studied form of synaptic plasticity, was discovered in the structure. So it seems to be able to put down a trace for what's happened to you in one shot, and then basically train the cortex, which is consolidation, to kind of retain that information and make that a long-term memory.
Have you seen the movie Memento? Have you
Yes.
heard of it?
Yeah,
you've heard of it. Okay, so
Yeah.
this is a Christopher Nolan film.
Yeah.
Early Christopher Nolan film, long before the Dark Knight series.
Yeah.
And it was about a guy who through trauma had anterograde amnesia, and the whole movie plays itself backwards in 5-minute scenes.
Yeah.
And the story is that I think someone had killed his wife, and he was trying to figure out who it was, and yet he couldn't remember what had happened 5 minutes before.
Yeah.
So it certainly takes some liberties with that film, but it's a great movie.
It is a great movie, and there are other sort of a lot of sort of famous cases of this type of amnesia, which really
Right.
show you what hippocampus does.
And so one very practical one. So for when you look at as a student looking at this circuit, I think what I would like you to know is that it's complex. It's intimately linked with the memory and emotional circuits are intimately linked. And if there was one area I'd like you to know about is probably the mammillary bodies and right here. And the reason why that is because these mammillary bodies are knocked out in severe thiamine deficiency, and this is quite common. As a faculty at Hennepin for many years, it is not unusual to get alcoholics come in confused because of severe thiamine deficiency. And you can actually convert them into a temporary problem into a permanent one if you accidentally give them glucose right away, because for reasons I could not cite you right now, you can actually turn it into a permanent problem. What you're seeing is an MRI scan, and what you're seeing here in the mammillary bodies, you get a T2 signal change, and that's what's essentially being shown here. Alcoholics are prone to all sorts of nutritional deficiencies because, of course, they don't vitamin supplement Phillips vodka. And if an alcoholic were to drink 750 milliliters of distilled spirits at 80 proof a day, that's 2,250 calories. They don't eat anything else. Wow. And so this is quite common. Emergency departments usually do a very good if there's anybody who comes in confused. In general, it's a good idea just to give them a blast of thiamine just in case. And
is this a reversible?
If they give it thiamine to them quickly, within a day or so, it's reversible. Yes. Yeah. All right. Next. Okay. So now we've done memory. We've done a patient's awake. We've tested their memory. Now we're going to test their language centers. And when I think about language, let me take off this screen here. So Matt, when I think about language, I think that language helps illustrate a simple principle I think of on occasion, which is, in general, the farther forward anterior you are in your brain, the more likely the brain is controlling efferent activity. Yeah. Right? And the farther posterior you are in the brain, the more likely it's sensory. Yeah. I think that's a good description. We always talk about the central sulcus as dividing the cortex into basically a sensory half and a motor half. And if you think about prefrontal cortex, ultimately it's just a complicated version of action planning. You heard it there from the expert on executive functioning. All right, next. So taking that into consideration when we're looking at language, and when you're testing someone's language, is to recognize that there are two large, relatively large regions of the cortex that help control language. They're unique in terms of cortical function in that you can really lesion them and see a problem. A lot of the cortex, which is very important, you can lesion, and it's quite difficult to kind of realize what you just did, right? Unless you're looking very closely with rather sensitive tools. But the Broca's areas is anterior in the frontal lobe, and not surprisingly, that is the region of expressive language. And the Wernicke's area back at the junction of the temporal parietal lobe is the region of receptive language processing. And there is a fasciculus of track of white matter projections that connect these two together. Right. Would you agree with that?
Absolutely.
Okay. All right. So how this is relevant is that these regions of the brain are frequently the site of lesions, whether or not it's a stroke, it's a brain tumor, whatever. And so you can end up with specific deficits related to these. And if you have Broca's aphasia, or a lesion in Broca's area, you get what you would expect if you're thinking about a damage to the expressive region of the brain, in that they will be unable to form words properly. You'll listen to them, and the words don't make any sense. They're non-fluent. And here's probably what's most relevant when you're actually looking at them, is that they look frustrated. You know, they just they want to say something, but they just can't get it out. Conversely, somebody with Wernicke's aphasia will just start talking. They'll talk non sequiturs. They'll just keep going and seem to be blissfully unaware. They might start to get agitated with you that you don't understand, that you clearly are not going along with whatever they're trying to communicate to you. But as far as they're concerned, they're kind of blissfully unaware of their deficits. One of the worst cases I had in the hospital was someone who had a stroke in Wernicke's area. And if you think about, language is so much more than just speaking, right?
Yeah.
It's nonverbal, it's being able to receive
Absolutely.
language, all sorts of different forms of language. So anyway, he woke up at 2 o'clock in the morning. He was confused, and nursing staff tried to calm him down.
Right.
Nursing assistants tried to calm him down. Residents tried, and
Couldn't.
he didn't get it. I mean,
Yeah.
he didn't, and not only when you're confronting someone, or someone feels like they're being confronted, there's all sorts of different ways that you can, take a body posture that's non-threatening and back away and give them a clear avenue to the door. These sorts of things. If you have Wernicke's aphasia, that doesn't make any sense. You're not picking up on any of that. All you just see are threats. And so anyway, he ran down the hall and was grabbed a fire extinguisher and started swinging that. Of course, security was called. They threw him down. He ended up with a spiral fracture. Which is a spiral fracture is a classic fracture seen in child abuse. It's essentially it's a torquing maneuver. I shouldn't say only child abuse, but it's almost universally seen in the setting of child abuse.
Yeah. So just to add, and maybe the way we sort of think about it in lectures, Wernicke seems to be involved in semantic processing, being able to sort of retrieve the meaning of words. So on the productive side, it's kind of a word salad, right? They invent words.
Neologism,
right. And then it's so garbled in the sense that the semantics or the meaning of the speech is kind of lost. And it's also is it true like for following instructions, they have difficulty following instructions?
Oh yes.
But Broca's wouldn't. You could tell them,
Yeah. Oh yeah. You just close your eyes.
Right. And they do this.
They'll close their eyes. Yeah. Hold your hands up. They'll hold your hands up.
Yeah.
In many ways, you really, when you have someone who's delirious, you need to tease out if they don't have a receptive language deficit.
Oh, okay.
All right. Okay. Next. Crane. Okay. Now we're moving down. We've jumped. I've insulted all of these people who work with the cortex. And we're jumping right into the cranial nerve. So going and thinking about the cranial nerves that are easily tested at the bedside, visual pathways are really helpful to think about. And this is a pathway where it's worthwhile to actually take a few moments to think about it. So what we're looking at here is in green are all of the information that's coming from the left visual field. I'm sorry. This is where it gets confusing because I'm sorry. We're looking at it from the head down. So I'm sorry. So green is the right visual field. Purple is the left visual field. So we're looking at this brain from the feet up, not from the top of the head down. All right. Sorry about that. And so the green information ultimately will end up, the information ultimately ends up on the opposite side.
Right.
Ultimately. And another way to tease that out is the information that comes from the inferior part of the visual field, so down lower, will end up, at least at some point in the radiations, ends up higher, and information from high in the visual field ends up radiating lower. It's a great example of how you go by the rule of opposites here, and what we're talking about here. So is if you take a look at here we are in the right visual field, information in the green goes into the retina and hits the retina on the opposite side. It then that green information travels through the optic nerves, and it stays monocular in the optic nerves. And then once it hits the optic chiasm, there's this very clever little switch that happens, and the temporal information, so the visual information from your peripheral visual fields, laterally anyway, crosses to the other side. This is particularly relevant because you can absolutely get lesions right here and see people arrive with bitemporal hemianopia. And then, but once you get past the optic chiasm, it's rather straightforward. A lesion will result in a homonymous hemianopia, meaning that what is happening occurs in both eyes, and both visual fields are the same. And then once you get through the optic nerve, past the optic chiasm, and then you end up getting into the lateral geniculate. And it is in the lateral geniculate that then there's a series of radiations that go back to the occipital cortex. And there is sense to this madness in terms of the radiations, right? So the higher the radiations go, and this is just two-dimensional, so it's hard to see this, but if these were to go superior up into the parietal lobe, those are almost always inferior visual fields. And the ones that go down lower into the temporal lobe end up being the superior visual fields. So I have another slide here that kind of shows what the lesions look like when this happens. Again, just to confuse, sorry about the confusion on this, but this is an example when you're looking at the head from the top down, not from the bottom up. But so right side visual field, left side visual field. So if you lesion the optic nerve, that's just going to be a monocular vision loss. If you lesion the optic chiasm, and a classic example here would be some mass that's emanating from the hypothalamus or from the pituitary, because that's the territory of the regional real estate that we're talking about. And that can cause a compression here of the optic chiasm, and you end up here with a bitemporal hemianopia. Not the person you want driving your car as you need to merge into traffic. And then as you go past that, now all of a sudden, once you're distal to the optic chiasm, any lesion, any complete lesion anyway, is going to result in a homonymous hemianopia of that other visual field. Once you get past the lateral geniculate, if you just get a series of striations, you can get these quadrantinopias, as we talked about before, related to how low or high the radiations are. One final note that I could imagine being on a step 1 examination is that if you have a homonymous hemianopia with central sparing, meaning that you still have central vision preserved, that usually means that the lesion was at the level of the occipital cortex and not in the optic radiations. And the reason why that is that the central vision has collateral vascularization. That would make evolutionary sense, that we doubled up our vascular perfusions to the critical areas of central vision. All right. So moving past vision, then the next thing we often do when we examine someone is look at their extraocular movements. 3 cranial nerves help control extraocular movements. Cranial nerve 3 does almost all of them, except for the lateral rectus, which is the 6th cranial nerve, the abducens nucleus, and cranial nerve 4, which serves the superior oblique. So if you have a cranial nerve 3 lesion, you end up over here, as these are all cranial nerve lesions on the right side. So cranial nerve 3 lesion, you lose all of your innervation except for the lateral rectus and the superior oblique. And so you end up with an eye that is everted, depressed downward. There is ptosis because, of course, cranial nerve 3 also helps hold up the levator palpebrae, right? And the cranial nerve 3 controls the parasympathetics, which helps constrict the pupil. So you end up with a blown or at least big midriatic pupil from a cranial nerve 3 lesion. Cranial nerve 4 lesions are often quite subtle. They really tend to look like the kid down here as opposed to the person up here. And that is because the superior oblique, the main purpose of the superior oblique is just to help us look at our nose. Is the way I've always somewhat thought about it. It helps our eyes look down and helps it internally rotate a little bit. So if you lose cranial nerve 4, you are not able. I said cranial nerve 4 before, didn't I? Okay,
sure,
good. All right, good. Please, you correct me
Yeah,
if I get this wrong.
think that's,
With cranial nerve 4, it helps us look at our nose. And so if you have a cranial nerve 4 lesion, what happens then is that eye then descends laterally and up. And if you think about it, so if my eye here had a cranial nerve 4 lesion, but I wanted to look at something, I would just compensate that with a head tilt. And so they walk around actually with their head.
Wow.
They can still see and they do this without even thinking about it, because otherwise they have rather profound diplopia. So that's a classic cranial nerve 4 lesion is a head tilt. Cranial nerve 6 is a little easier to comprehend. It's just the lateral rectus. So if you have lateral rectus palsy, you can't abduct that eye, and you can end up with an eye that looks a little bit like this. Oftentimes it's more subtle than that, but this is a nice example of it. Jumping on, so to state the obvious, Matt, eye movements are complicated. They're fascinating. Have you studied eye movements much?
We do study some eye movements, sort of cortical representation of eye movements. It's interesting. There's at least 3 different cortical eye fields. A lot of cortex has evolved to control eye movements because for humans, with foveal vision, it's critical. Where you look controls what information comes into your brain.
It is,
and so
it is stunning. It is stunning.
Yeah.
Just the brainstem alone control of eye movements is pretty fascinating.
Absolutely.
So we are not going to get into all of this, but I just want you to appreciate it the next time you're on the street and see a car go by and then just suddenly follow that car. That is a smooth eye movement. And then as you're following that car go by, and then your vision suddenly shifts to a friend walking down the street. That is a saccadic eye movement. So the coordination that would cause your vision to slowly move to the left side is a very different circuitry that causes your vision to suddenly move to the left side. And then to allow even more beautiful complexity to it is when you make that sudden saccadic eye movement, is that it's a different group of neurons which start the saccadic eye movement, which maintain it, and then stop it.
Yeah.
I mean, it's just like, really? I mean, that's just astonishing. Okay.
Yeah.
So all of that being said, most of that you will not be able to test readily at the bedside, but there is one thing that is particularly useful, which is testing conjugate horizontal vision through the medial longitudinal fasciculus. And there's a reason for this, which is that the tract that controls that is the medial longitudinal fasciculus, which is fairly long. It goes in as terms of brain stem circuits go, it goes from the bottom end of the pons all the way up through the pons and well up into the midbrain to link cranial nerve 6 and cranial nerve 3 together to help create that smooth horizontal eye movement. And this is not a terrible representation of what this looks like, but what we're looking at here is what happens when we want to look to the right side. Okay, so you generate that signal from your left-sided cortex. And this is, of course, when we cortically do this. This happens subconsciously all the time due to our vestibular nuclei. But let's say you wanted to decide that you were going to look over to the right side. Your left cortex will send that signal. Left frontal cortex, remember, frontal part of the brain helps move things. And then that sends a signal down, descends and crosses over into the paramedian pontine reticular formation, these bilateral blue structures. And the paramedian pontine reticular formation on the right helps us turn our vision to the right, and the paramedian pontine reticular formation on the left helps us look to the left. And it makes a lot of sense because the PPRF is right next to abducens, right, which moves it in that direction. However, in order to link the abducens on one side with the medial rectus, i.e. cranial nerve 3, on the other side, you need this long circuitry, which is not really described as long here, but it's pretty long, okay? And this is the medial longitudinal fasciculus right here. It decussates immediately over the other side and climbs up. This region right here, this medial longitudinal fasciculus, is prone to injury. You can get it in a stroke, and in particularly you can get it in demyelinating disease because it is, after all, white matter. So in multiple sclerosis, we see this fairly frequently. And so this is another example of what it looks like. Here's the frontal eye field on one side, sending a signal down to the other side, to the paramedian pontine reticular formation, where it sends a signal to that side's sixth to pull the eye over laterally to abduct the eye. And then it also signals immediately decusate and go all up and down this medial longitudinal fasciculus. And so if you have a lesion right here, or here, or here, or here, or here, you will end up with the same picture, which is an eye. So I'm gonna try and move to this side here. So this eye will perfectly fine AB duct that way, but this one just stares out straight.
Right.
And so that's called an intranuclear ophthalmoplegia, and we'll see that in demyelinating disease in particular.
Yeah, I guess it can be bilateral or unilateral. Is it?
Right. Yeah. So especially if it's bilateral, you can get a lesion right there. If it's bilateral, you almost don't even need to do an MRI scan.
Right. Yeah.
I mean, it's 2018. We always do an MRI scan. Yeah. But
Yeah.
But
it's so obvious clinically that
that is a sine qua non ipso facto. If you got that, it's almost certainly a demyelinating lesion. And one of the reasons for that is because it can't be a vascular lesion because the vasculation respects the midline.
Oh, okay.
Right?
Interesting. Yeah.
So anyway. Okie dokie. Next. Cranial nerves. So cranial nerve 5 does a lot. All you have to do is go back to your netter. There's all of the various different purposes it serves. Chewing motions. What is very practical just at the bedside though is just to look at the sensory function. So cranial nerve 5 serves the sensation to the face, and is in 3 distributions: the ophthalmic nerve, maxillary nerve, and mandibular nerve. There is a very common in my world anyway, in the world of neurologists, trigeminal neuralgia, where there is a sudden lancinating pain, usually in the V2 distribution, and it's due to irritation of the trigeminal nerve. It can be quite severe. What's so frustrating about it, people have very little control over it. It'll just happen. Sometimes just shaving, wind blowing on their face will set it off. And so that is and just testing pinprick sensation over the face will be helpful. Interestingly enough, and this will get when you think about, we're jumping ahead a little bit, but it's useful. When we think about sensory, think about pain, think about vibration. Not that proprioception, soft touch, temperature. Not all those things have their place. But pain and vibration are very easy to test at the bedside. So when you want to test the face, you take pins, safety pins, and go over the face, make sure they can feel it. If you want to test vibration, you really can't test it very well, because if you put it over the skull will vibrate to the other side, and the other side will pick it up as well. So mainly just pain over the face. All right, next. After the signal has come in, how does the afferent information get up to the thalamus? And this is, I got to tell you, it's
It's complicated.
it's complicated. I still like trying to remind myself, what's the mesencephalic nucleus of 5 all
Sure,
about?
yeah.
What's the primary nucleus of 5
Yeah.
all about? Here's what I think is useful to know is that a lot of trigeminal afferent information goes down through the trigeminal spinal thalamic tract. It
Right.
makes this, it doesn't just go in and then go up. It makes this unusual kind of loop down into the medulla, sometimes all the way down to the cervical spinal cord, and then it loops back up. Why is this relevant? The reason why this is relevant is that you can, as we'll point out here in a minute, you can have a lesion down here in the medulla, and it manifests itself with loss of pain and temperature on that side of the face.
Right. So that's mostly pain and temperature that goes to
Yes.
the spinal nucleus
Yes.
of 5.
The
right?
yep. Okay, moving on, jumping on to cranial nerve 7. All right, so there's an anomaly. Is that how you described it? Kind of an anomaly?
Yeah, or just it's just an oddity of evolution that this particular combination of projections developed. But as you've said, we'll show. It has very clear clinical
It has
significance.
really clear clinical significance, which is that, so the upper part of your face is the efferent information, the motor information to your forehead has bilateral innervation.
Right, from motor cortex down to the facial nucleus.
Yep, but the lower part of your face, just contralateral innervation.
Right.
And so why is that? And why is that important? It's important because if you have a stroke or a demyelinating plaque or any lesion of the cortical spinal / cortical bulbar tracts above the cranial nerve 7 nuclei, you get this very specific pattern of weakness, which is they're just weak.
Right.
They're just weak in the lower part of their face, but their forehead's fine.
Right.
Conversely, if it's the cranial nerve 7, it's the whole, the eyes involved, and which is interesting because it's one of those situations where the more severe the motor deficit, the less you are concerned about a bad stroke. Which is kind of, people often think is like, oh my, people are terrified when
Right.
they can't move their
Oh,
face,
oh,
right?
yeah.
I mean, that's where you go to smile, right?
Right.
Smiling's best.
Right.
Something else about cranial nerve 7 just to think about is if you're trying to tease it out, is this a stroke or is this a cranial nerve 7 lesion, is that people with Bell's palsy, this is just another schematic that shows the same thing, people with Bell's palsy, which is a cranial nerve 7 lesion, have other problems that are just served by cranial nerve 7. Taste, for example. They can't taste on the anterior 2/3 of that side of their tongue. They have hyperacusis because one of the first branches of cranial nerve 7 is the nerve to the stapedius, which helps dampen the tympanic membrane in loud environments. All right, cranial nerves. Okay, now as we're going down and we're looking at palate elevation, having them say ah, shoulder shrug, and tongue movements. And palate elevation is cranial nerves 9 and 10, shoulder shrug is accessory, 11, tongue movements is the hypoglossal at 12. So let's just think about classic step 1 question. They either tell you there's a lesion of a cranial nerve on the left side, or they show you a picture and they ask you which side is the cranial nerve lesion on. So if you have a left-sided lesion of cranial nerve 9 or 10, which helps elevate up the palate, what will happen then is that the opposites, the uvula will deviate to the opposite side. So here we are. This is a situation where we're imagining a left-sided lesion to cranial nerves 9, 10, or maybe both of them, and they're not able to raise that part of the palate, the levator veli palatini and the tensor veli palatini. And yes, I did have to look that up before. And so what you'll see is the uvula deviate over to the contralateral side, which is interesting in that the picture looks exactly the opposite when it's a tongue movement problem. So because what happened, the hypoglossal nerve on the left side moves the tongue to the right side, and the hypoglossal nerve on the right side moves it to the left side. So if you have a left-sided hypoglossal nerve lesion, you will get the tongue deviating to the left side here, as Hannah Montana here nicely demonstrates. Okay, we've done all cranial nerves. We're rolling right along. If there was one thing, if there was one track to know, this has got to be it.
Yeah.
I think.
Yeah.
This is where the money is. This is the money shot. In particular, so the cortical spinal tracts, but particular 95% of your energy should be on the lateral cortical spinal tract.
Yeah.
There is a medial cortical spinal tract. Brent Clark has shown it to me on slides. It is undoubtedly fascinating and cool and all of that. Clinically relevant, in terms of bedside evaluation, not so much. So these are the motor tracts. The pyramidal neurons. Pyramidal neurons are just kind of amazing, aren't they? So
yeah.
you have these, and they look like little pyramids, right? You have
They
these
do.
little pyramids up in the motor cortex, and they descend. One neuron, one cell descends all the way down, decussates over, and then lands in the spinal cord.
Yep.
I mean, that's astonishing.
It is amazing.
Yeah, and we're learning how to regrow those, which is totally cool. All right. So anyway, in terms of the localization here, though, so you have the cortical spinal tract pyramidal neurons. The signal comes from the motor cortex, descends. A couple important landmarks here: cerebral peduncles, pons. Call that basis pontis. How would you?
Pontine fascicles.
Okay.
It just sort of breaks up into these small bundles of axons that you can have seen in lab and carry the same information down.
Excellent.
Before reaggregating. Yeah.
And then here is the landmark to know where it crosses, right? It crosses at the pyramids in the medulla, the pyramidal decussation, and then it crosses over into the lateral funiculus. That's where the name comes from, lateral cortical spinal tract, and then ultimately ends up on an anterior horn cell.
Right.
Okay. If this is all you got, if this is all the bandwidth, if you only have the bandwidth for one track, this is the one you got to know. So and the reason why is, if there's a lesion anywhere here, what is that going to look like?
Yeah.
Next, so sensory. So as we talked about before, the two ways to think about it are the you want to test pain because it's easy to test, and you want to test vibration because it's easy to test. Not that temperature isn't important. Interestingly, when people think about which is more important, pain or vibration, they'd probably say, well, I want the signals for pain to get to me fastest,
Yeah.
right? But that's not the way.
Yeah.
That's not the way evolutionists kind of figured this out. And if you spend any time thinking about it, you really want proprioception to be lightning fast so that you don't fall over as you're walking.
Right.
This is why if you touch something hot, it's like, well, maybe that's not. Oh, yeah, that's because it's unmyelinated. It's unmyelinated or very weakly myelinated axons. When meanwhile, vibration and proprioception are just like these big fat. Big, beautiful myelinated axons to just zipping saltatory conduction, right? How did I do on that?
Perfect.
Did I do that? All right,
Absolutely.
good. All right, so here we are. So now as you're thinking about the lateral spinal thalamic tract, we're talking about information going the opposite direction, right? We're not talking top down. We're talking about information that comes in through the dorsal root, crosses over nearly immediately. Yes. Some of you may remember that there's this information can cross one or two levels above where the input goes in, but for all practical purposes, this information decussates almost immediately after entering the spinal cord, and that means that your pain sensation for the left side, for your left leg, is traveling up nearly the entirety of your right spinal cord. And then it goes up into the ventral posterolateral nucleus. And do you have a little mnemonic for, not mnemonic, but acronym for remembering that?
I don't.
I do.
Oh, what is that?
I do. So very painful leg, VPL.
Oh, nice.
Not to be confused where the trigeminal
Right.
spinal tract goes, which is the VPM,
Yeah,
or very painful mouth.
perfect.
Yeah. So there you go. So
Yeah.
pain from the
great.
extremities goes to the VPL, ventral posterolateral nucleus, or very painful leg, and the pain for the face goes to the VPM, very painful mouth. All right. Moving on. Very different tract though for the afferent information from vibration. Oh, this just shows exactly what I was telling you before,
Right.
which is that this is the trigeminal painful circuit.
Spinal nucleus of 5.
Thank you. Spinal nucleus of 5. Thank you. Which ends up at the VPM, and the lateral spinal thalamic tract ends up at the VPL. All right. Sensory. So what about vibration and proprioception? The input also goes in right through the dorsal root ganglia. It ascends immediately up into the dorsal columns, i.e., where it gets its name, and then it decussates kind of about at the same level where the motor tracts decussate but go in the other direction.
Right.
And then it crosses over into the medial lemniscus, which is medial to pretty much everything else. Pretty much the other two tracts that we were talking about. And again, also ends up at the VPL nucleus of the thalamus. So important regions to know: dorsal columns, decussates in the medulla, is then the medial lemniscus. Probably want to know the difference between the nucleus gracilis versus the nucleus cuneatus.
Right.
That's a step one question somebody would
And
ask.
Right.
Yeah,
and then there's a relay in the dorsal column nuclei.
Okay,
Before
yeah, tell us
decussates,
about
right?
that. Yeah, tell us about that.
Yeah, so the fibers come in from mechanoreceptors, for example, and
Yep.
do just exactly what Mike
Right-
was saying. They
right
ascend,
here.
yep, and without synapsing, they enter and ascend in the dorsal columns, and then the first synapse in that pathway is in the dorsal column nuclei, the nucleus
Right here.
gracilis,
Nucleus
right,
gracilis,
purposely-
nucleus
purposely
cuneatus. Thank you. Yep.
cuneatus, Thank you. And
Yep.
then the axons of those relay neurons then decussate and then ascend.
And then they go up.
And then they go up.
So second order neurons,
Second
and,
order, yep.
and then they land in the thalamus, where the third order neurons go all the way up to the
Right.
tippy top.
Yep.
All right, great. All right, and here's just another picture of the same thing. You get yourself a handy tuning fork, very easy to test. Cerebellum, you can let yourself get completely tied up in numerous complicated circuits. It is, so the cerebellum helps promote smooth movements by constantly providing afferent information back to the motor circuit to allow for precision. Okay, and it's needless to say it's quite complicated practically, and so I just threw a schematic up here just to give you a little bit of an idea of how deep. How the beginnings of what a deep dive into the cerebellum would look like. Here's what I think you need. Here's what I think you really need to know: is that cerebellar lesions nearly universally manifest themselves unilaterally. So if you have a lesion in the right side of the cerebellum, you will have right-sided ataxia. If you have a lesion in right-sided cerebellar tracts, you will have right-sided ataxia, and so on. And I really, it's, short of becoming a movement disorders expert and dealing with people who have unusual neurodegenerative syndromes, I think that's fine. And I can't imagine a step one would really go into much more detail than that. Let's move on to another complicated system. Here's a system where I think, a little different from the cerebellar system. I think it still does behoove us to spend a little bit of time looking at the various circuits, and so that we get our heads around the extrapyramidal circuit. So the extrapyramidal circuit, like the cerebellum, does not directly innervate anterior horn cells to move muscles, but instead they provide input and modulation to the cortex so that movements and I'll point it, and not just movements. The extrapyramidal circuit does other things besides just movement, but movement is so easy to test. That's what we always look for. One way, so there's a direct pathway. There's an indirect pathway. It is worthwhile to actually go through it and become familiar a little bit in terms of, okay, the cortex activates the striatum, which inhibits the internal globus pallidus, which, and on and on and on. A couple large order ways to think about it is that the direct pathway, well, let me back up. A good way to think about it is that for proper movement to happen, you have to have a balance between an activation, an overall turning up the volume of the motor system and a decrease of the motor system. It needs to be. I mean, again, this is kind of what's so amazing about the nervous. I mean, like all this stuff just happens for us, right? And so it's kind of when it works well, it's kind of perfectly in balance with each other. But what happens when it doesn't go well? You end up with either hyperkinetic motor problems or hypokinetic motor problems. And so if you think about the two different pathways, is that in general, the direct pathway facilitates, is part of what's driving is turning the volume up in terms of motor activity through the cortex, and the indirect pathway is turning the volume down. And when they work well, they're kind of just perfectly in balance with each other. Okay, so let's say they don't work well. All right. So what about if you have a lesion to the subthalamus, which can happen in stroke? We also use the subthalamus as a stimulating target in Parkinson's disease and other movement disorders. What happens then in that part of the circuit is that you end up with a unilateral hyperkinetic movement disorder. Or if you happen to have both of them knocked out, you'd end up with a bilateral hyperkinetic movement disorder, and I have a video in a second to show you what one of those looks like. Conversely, in the setting of Parkinson's disease, where you have the substantia nigra compacta, that's a step one question right there. Compacta, not
reticulata.
Not reticulata.
Yeah.
Which is also part of the whole circuit too, right? You can end up with and because Parkinson's disease, Parkinsonism usually manifests itself usually bilaterally. Parkinson's disease often manifests itself initially in one arm, but that doesn't mean the other arm isn't affected. And so then you end up with a hypokinetic. I have a couple of videos here, but Matt, I know you do kind of some explanation of the extrapyramidal circuit. Any other kind of thoughts on how you would kind of?
No, I mean I think that's about right. Typically, the way we're describing it is as really sort of a gate on motor output that you have to open in order to move, and sort of thinking of that because the output of the basal ganglia on the motor thalamus is inhibitory.
Okay.
And it's sort of those output neurons clamp the motor thalamus down. You got to open the gate to move, and that's sort of the direct pathway, and the indirect pathway closes the gate again. And so damage to one or the other aspect of the circuit produces hypokinetic or hyperkinetic disorders. Okie
dokie. So where we are now is I'm just going to show two examples of one, a hyperkinetic movement disorder. This is a woman who had a stroke to the subthalamus, and she has hemibalism. And you get an idea of what that looks like here.
And then put your arms out in front of you.
So this lesion is in her right subthalamus.
Lay down again.
And so she has
and try with the left hand.
hemiballistic movements of her left. That's kind of amazing.
That's good.
So her gait is actually pretty good.
And back again.
But look what happens with her arm. Look at all the extra movements you get with her arm swings. Now, if we then go to Parkinson's, please.
So and that just fits with what you were saying before, because the subthalamic nucleus is part of the indirect pathway that would normally inhibit movement, and so if it's lesioned or damaged, sort of gate fails open and too much activity is coming through and too much movement is being produced. So it kind of fits with the circuitry of
That makes sense.
of the indirect pathway.
That makes sense. So when most people think of Parkinson's disease, they usually think of Michael J. Fox or a tremor disorder. I'd ask you, instead of thinking of Parkinson's disease as a disorder of tremor, think of it as a disorder of slowness. Everything just slows down. Gait slows down, facial movements slow down, arm swing slows down, handwriting gets small, it takes forever to eat. And so here's just an example of someone with Parkinson's disease and their gait. And you'll notice the difference between when they're on medications and off. So look, very little arm swing, no arm swing really, stooped posture, and look at the difference. Arms swinging back and forth, more animated. Nice example of Parkinson's disease. All right, now let's just do a little bonus for vasculature orientation. So we're just going to go from the bottom up, starting first with the vertebral basilar system. The basilar artery is formed by two vertebral arteries, and out of those vertebral arteries are little penetrating arteries that serve the anterior portion of the medulla, in addition to a fairly large and important artery called the posterior inferior cerebellar artery. And so what we're looking at here is vascular territory, and you'll see quite a bit of the posterior inferior cerebellar artery, and you can't see it here, but the pica also serves the lateral medulla, and so you can often get a lateral medullary syndrome. And this is very common in the setting of a vertebral dissection. So vertebral dissection is a form of stroke that often happens in the young. They don't yet have atherosclerotic disease, but they have vessels that can tear spontaneously with sneezing, coughing, whiplash, motor vehicle accidents, straining. And you can, as the vertebral artery tears, it can occlude the posterior inferior cerebellar artery. And we're not going to go through all the tracks again, but let me just assure you that a few things that flow through this area of the lateral medulla right here include the ipsilateral cerebellar findings, so they'll have ataxia on that side. They'll also have those descending fibers from the spinal trigeminal tract and nucleus go down, so they'll have ipsilateral loss of pain on that side. However, they have the loss of pain and temperature on the opposite side of their body because the lateral spinal thalamic tract is running through there. And so you get this crossed sensory loss, and that is very consistent with a pica infarction. Basilar artery, I think all of us take a moment here just to appreciate how important our basilar artery is. You can have infarctions there, tragically. And if it's a big infarction, you lose all motor function. You may or may not lose sensory function from the extremities. You lose, if it's higher than cranial nerve 7, you lose all facial movements, and you also lose horizontal. That's correct. You lose horizontal gaze because of the paramedian pontine reticular formation and both medial longitudinal fasciculi. But what you do have is you still have vertical eye movements, the eyes that can go up and down, and they are people when this happens, they are conscious. Yeah. And so this happens when I was faculty at Hennepin. I remember a new patient we had came into the emergency department, and the story was he was just suddenly unresponsive. And I went in, and he wasn't unresponsive at all because he just looked. He just looked at me just with a look of sheer terror, and was able to move his eyes up and down. And that's a locked-in syndrome. Moving right along. You can another classic stroke syndrome that you can have involves cranial nerve 3, as well as that the cerebral peduncle on that side, and that can create this very typical picture of hemiparesis with facial droop, just of the lower face, of course, on the contralateral side, and yet ipsilateral cranial nerve 3 palsy. And so this, you'll have an ipsilateral palsy here, and on the other side of the body, you'll have the weakness. And in general, if you're dealing with a stroke syndrome, if you can find a cranial nerve sign, that tells you what side of the body it's on, because it's on that ipsilateral side, just as a general rule. Next. Okay, now we're all up. Now that we've appreciated our basilar artery, let's appreciate our circle of Willis, the blood beltway to the brain, so that if we happen to get a major stenosis in one of the 3 main arteries leading to our head, the right and left internal carotid arteries here, or the basilar artery, the rest of the vasculature can perfuse, provide collateral circulation. And so we're going to talk just a little bit about what the big obstructions of the big arteries in the cerebrum look like. So once you get up above the brainstem, you're talking about the anterior cerebral artery circulation, middle cerebral artery circulation, and both of these come from the internal carotid artery, and then the posterior cerebral artery, which comes from the basilar artery, plus the circle of Willis. All right. This is a picture you'll see frequently, but just recognize that the vascularization on the medial aspect of the brain looks quite different, in that the anterior cerebral artery serves almost all of midline structures of the cortex, except for the occipital lobes, which is mainly the posterior cerebral artery. That is relevant when it comes to signs for stroke. So let's just go quickly over what these strokes look like. So if you have a middle cerebral artery infarction in the dominant hemisphere, which is almost always the left hemisphere, even in left-handed individuals, it's often the left side. You do need to ask though about handedness, just so at least it makes you think about whether or not you might be dealing with a different, typically dominant hemisphere syndrome. All right. So if you have a typical right-handed individual, and they infarct out their middle cerebral artery of their dominant hemisphere, they will be aphasic, and they will have contralateral. They'll have contralateral weakness of the lower face and the hand, but the leg is fine, and that's really important to understand why. Real quick before we get to that, what if you have an infarction of your non-dominant hemisphere? Then, in addition to having lower face, hand weakness, normal leg, they have neglect. And neglect is one of those super cool things to learn about, and you also see it on exam. It is one of those it's one of those things that we can actually pick up on examination, because they will not attend. So if I lost my non-dominant hemisphere, I would not attend to this side of the world, and you can get the alien hand syndrome, that people get kind of handsy on this side. That isn't nearly as common, but what you can do is you can just do sensory. You do bilateral sensory stimulation. So if I'm neglecting this hand, I stimulate this hand, I say, yeah, I can feel it. I stimulate this hand, I say, yeah, I can feel it. But if I stimulate them both together, they're like, yeah, this is the only one I feel. So it's a common, simple bedside test of neglect. All right. Okay, so here's why we don't get that leg. This is the cortical. Can you talk about how a homunculus, it's cortical mapping on how we kind these things
basically
out?
It was sort of discovered by electrical stimulation that if you surgically expose the precentral gyrus, the motor strip, you can stimulate this, and then when you do, these different parts of the body twitch, which sort of read out the wiring from motor cortex to the musculature. And the homunculus is, it's somatotopic in the sense that the the relationships between the body parts is preserved really right on the cortical surface, as you see.
So yeah, so a lot, and this also tells a lot of what the brain values.
Yeah.
Right. So the brain loves the hand.
Yeah.
Which makes
Absolutely.
sense.
Yeah.
It loves the face.
Yeah.
And in particular, I think on the sensory map, I don't know if I have the sensory map on here or not, but the face is big on the sensory map. In particular, like this finger.
Yeah.
Right. I mean, like the cortex loves this finger above all others.
Yeah. Yeah, and probably the reason for that is, these are the most complicated parts of the body to control. They have the most degrees of freedom, the most small muscles. And you can think for humans how important, hand motor control is. This is sort of, it's sort of the origin of all tool use. This is the unique human motor specialization, that and speech. So my understanding is from motor
being able to throw things, it's just
tool
incredible.
Use. And so since so much cortex is devoted to these structures, motor deficits in them are particularly pronounced following stroke.
All right, so so if you have a middle cerebral artery infarct, you will lose all of this territory here, but your leg will be okay.
Yeah.
Or if you have an anterior cerebral artery infarct, you'll lose all of this, but the rest of this will be okay. So what happens if you don't have a big cortical infarct, but you just have a small vessel infarct, usually due to hypertension, diabetes. And so this is an infarct of the right. Again, this is a little confusing. This is the right side of the brain. This is the left side of the brain. We are looking from the feet up at the brain here. So this is the right internal posterior limb of the internal capsule, which is where the spinal cortical bulbar tracts pass. You can get very frequently, you can lose motor, face, hand, leg. So this is another example of the more severe the motor deficit, often the less pronounced the stroke, for example. So, I mean, you wouldn't be happy to have this stroke, but you'd be glad that you preserved your cortical function. If you, in a universe where you'd be able to have that option. What about the anterior cerebral artery? So this will, if you lose your anterior cerebral artery, this is an example of the left anterior cerebral artery infarction. You will lose those frontal eye fields that we talked about before. So the left frontal eye field helps you look to the right. So you will end up just staring off to a patient will end up being staring off to the left side. And usually how we look at this is that these people are often in the intensive care unit and sedated, but you just open up their eyes and they're just like, oh yeah, they got a gaze preference. Yeah. So what about the posterior cerebral artery? So you'll get, as we talked about before with the visual pathways, you'll get a contralateral homogeneous hemianopia, but often with central sparing. And you often will get agnosia as a syndrome that we do see if you look for it, that they're blind, but they are unaware that they are blind. That
is amazing.
Isn't that just kind of blow you away a little bit? It's just like, and the people will have, if they happen to have bilateral posterior circulation, I mean, they will be fully blind and genuinely deny that they are blind. I mean, it's just amazing. Isn't that something?
Yeah.
Is that the brain has to recognize it has a problem. Okay, we're nearing the end here. Let's just down into the spinal cord, because if you like anything that happens underneath your chin, you're a fan of your spinal cord. And it's pretty simple vascularization, but just remember that one anterior spinal artery provides most of the vascularization, except a little bit of the dorsal horns, the dorsal columns, which come from two posterior spinal arteries. Clinical syndrome where this is relevant is you can get a loss of the artery of Adamkowitz, which comes off of the aorta. And if you do that, you lose all of this territory. So you lose this gray matter, which serves that level, but you also lose cortical spinal tracts. You lose lateral spinal thalamic tracts. So you have bilateral paralysis. You have bilateral loss of pain and temperature, but you can feel vibration and proprioception still. So you got that going for you. This does happen with people who've had procedures done on the aorta. So like abdominal aortic aneurysm surgeries. which are challenge. If you talk to surgeons about it, they're challenging enough, let alone having to worry about this. And that is it. I want to thank Matt Chafee so much
Oh, it was a pleasure.
for walking us through neuroanatomy. Next on Brain Pods Volume 2 is going to be neurobiology and neuropharmacology. I'm looking for a very special guest, and hopefully we'll be able to announce her soon. All right, thank you very much, Matt.
Yeah,
Any
it's a
final
pleasure.
words?
No, that's it. I really enjoyed this, and thanks for the overview. It helps us think about focusing what we do in year one.
Well, yeah, and going back to year one is good for all of us too. All right, thank you. Bye. See you later.